Hello, everyone. Welcome to our symposium. This is Shao Zhenqin from Beijing Normal University. On behalf of Chair Professor Yuejia Luo, I'm going to highlight 5 talks in this symposium. These talks target on a newly emerging topic of computational psychophysiology, with a focus on human sleep, emotion, and cognition. The first talk was given by Yunzhe Liu. Dr. Liu was trained at University College of London and Oxford University. He then became a research faculty at UCL. He recently joined Beijing Normal University as a new PI at McGovern Institute for Brain Research. He has a talk will focus on experiences, replay, and companies' efficient non-local learning, meaning using MEG technique. The second talk was given by Xuebing Li. Dr. Li is an associate professor at the Institute of Psychology at Chinese Academy of Sciences. She will share her recent data on how emotional working memory training promotes explicit emotion regulation and implicit negative control. After that, Haiyan Wu, assistant professor from Department of Psychology at University of Macau, will give a third talk. She will share recent data from her lab about temporal dynamics of anniversary processing, evidence from mouse tracking, EEG, SEEG. We then move on the first talk by Frederick Weber. Dr. Weber works at the Dundas Institute for Brain, Cognition, and Behavior, the Netherlands. He has developed a sleep trip toolbox widely used in the field of human sleep research. He will present sleep trip as a tool to study clinical population. The fifth talk was given by Dr. Wei Wu. Dr. Wu is a researcher scientist at Stanford University School of Medicine. He has developed advanced machine learning algorithms for analyzing EEG data in clinical populations. His talk today will focus on towards machine learning driven EEG biomarkers for precision mental health. So I hope you will enjoy these talks and see you soon on our virtual conference.
So my name is Yangzi, and I'm going to talk about a recent work about linking replay to model-based learning. Cool. So now let's imagine if you are this rodent and the only thing you want is this piece of cheese, okay? And things might get a bit complicated in a real life situation. For example, you might start from all 3 locations. And again, the goal of your life is to get this piece of cheese. Now imagine in one situation, you start from the blue and you choose the left option and you get the cheese. That's great, right? However, if you want to be more efficient, you should also know what you should do when you are being put in the yellow or the green location, right? So that's saying I should know whether the other road is also leading to the chase or not. And this does create a problem, right? Because the state and action and then the outcome are separated by both time and space. And this is a well-known problem called credit assignment in the reinforcement learning. And today I want to show you how in the human brain could we implement a model-based learning algorithm that's trying to solve this problem. And I think one potential mechanism is replay. So just a little bit background on replay. So replay is a well-documented phenomenon in rodents. So the play cells that are normally only going to fire at a given location will fire sequentially during rest, that actually recapitulating past or future experience. That's what we call replay. It's a suitable mechanism because it is internally generated, so it can quickly, and it's also time compressed, so it can very quickly bridge action and space that are, the action and outcome that are separated by time and space. All right, so how we do that? So the first thing we need to do, we need to design tasks that dissociate local and non-local learning. And here is what I mean. So let's say, again, I'm in the blue path and take this one. And then the path I just chosen is what I call local learning, right? You can just do that through try and error. And there are two other paths, I would call that non-local learning. It's non-local because you have the lead by passing the local outcome to the non-local actions. And the way to do it is what I termed non-local learning. And if this, and upon this problem, there is a second order question, i.e. with so many non-local experience in our life, how should we prioritize? How should we prioritize the memory access? Right? Should I replay more of first to the path lead to yellow versus the green, or vice versa? Luckily, this problem has already been solved by Marcello Marta and Nathaniel Dahl 2 years ago, based on reinforcement learning. So they figured out a lumping model, i.e. saying the way to decide which memory should I have access to first, depending on a product of two factors. One is called need, another called gain. And need determines how often did I encounter this experience again. And the gain determines how much actual reward do I get by replay the experience. And with close collaboration with Marcello and Nathaniel, also with Tim Barons and Ray Dolan, we're trying to do that. We're trying to do that in humans. So in humans, this is the task they're doing. So we manipulate lead by gain separately. The lead is manipulated by the probability they're going to start at each of the arms. So at each trial, the participant be put in one of those arms based on those starting probability. And then the job of the participants is choose the path leading to the outcome, i.e. X and Y. And the reward probability of X and Y going to change independently, follow a random walk. So this is going to manipulate the gain. And the aim of this study, and then we can study priority. And then the aim of this study is to look at those triangles, i.e. priority, replay, and behavior. All right, so first let's talk about the model-based behavior. So we can define model-based behavior in a model agnostic way. So again, we already separate local and non-local learning. Let's say again, on the last trial, we start from the blue. And then in the current trial, if I have been putting in a different starting arm, if I am model-based, i.e. if I know the transition structure, I should more likely to choose this path versus this one, right? Because I already get a reward on this path. So this is what we can use as index of model-based behavior, while we can using some model agnostic analysis to detect that. And indeed, this is what we find. So the same means starting at the same arm as previous trial, diff means starting from different arm. So the reward means get a reward last trial, unarg means didn't get a reward. So this contrast is a textbook of model-free learning. So you do that through try and error. And this would be an index of model-based learning, i.e. if I starting from a different arm, what is the probability do I going to choose the path that leading to the same outcome as the last trial? And you would imagine if I'm model-based, I would more likely to do so if I get a reward on the last trial. And this is indeed the case. And the difference between model-free and model-based in this task are indifferentiable, which means the participants indeed are really model-based in this task. Then we ask a second question. In the model-based behavior, do we see prioritization? We can further separate each of the non-local paths as high lead versus low lead, or high gain versus low gain. If we compare the learning rate of the high high priority versus low priority, we see indeed a higher, significantly higher learning rate for the high priority path versus no priority. So this establishes the first line of this triangle, i.e. the behavior i.e. there is prioritization in behavior. All right, so let's now turn to the neural data. So again, so this task, we want to look at the neural mechanism. So first thing first, we want to say, do we actually can decode all those pictures? And this is not a trivial task because there are 16 states in this task, right? So because there are 18 pictures in this task because there are 6 sequences. Each sequence have 3 distinct stimuli. So first, we can show that we can decode all the picture pretty well with decoding accuracy peaked around 50%, while the chance level is below 10%. And again, peaked around 200 milliseconds, consistent with our previous study. And the second thing we want to look at is only look at the time when the reward has been received, because this is only time in this task where new learning can happen. And if we look at those time, we can say in general, we have 2 types of sequence. So the x-axis here are the time lag. The smaller the time lag means the faster the replay is. And the y-axis means the strength of the replay. And the dotted line is the permutation threshold. Anything that past that would be significant. So you can see we can identify 2 types of replay, one in forward direction. So forward indicating as the same direction as experience, while backward means in the opposite direction as experience. And it's interesting because this forward thing will replicate our previous study doing rest that we show a dominant forward thing at 40 or 50 millisecond time lag. So it's really, really fast. But this backward one is intriguing. It's slower, it's backward. It potentially may be related to non-local learning. Now, again, in this task, we separate local and non-local learning. So the first thing we want to say is, do we see a representation content difference in those 2 replay with respect to non-local and local representation? And indeed, we say this 30 millisecond forward replay mostly encoding the local experience, i.e. the experience you just go through. So it's indeed literally replay, while it doesn't represent the non-local experience. However, the 106 milliseconds show a distinct pattern. It only encodes non-local experience and doesn't care about the local one. And it's interesting. Then we can look at the physiological difference, right? Again, we show the 30 millisecond thing is associated with a shape width ripple power increase at 120 to 180. And this is consistent with our previous studies for the similar kind of replay. But we didn't see that for the 160 millisecond. And a few of the beamforming results, which I haven't shown here, suggest this 160 millisecond have a more cortical engagement as compared to the 30 millisecond. So this all of those indicating the 160 millisecond replay is another type of replay and is potentially suitable for non-local learning. And we're going to test that formally through a computational model. The idea is to say, does the inclusion of sequence going to significantly increase the learning, i.e. the learning rate we put on the replay, while already taking into account of the baseline that doesn't care about replay. And now we can say, do we say 160 millisecond increase non-local learning or 31 increase? So the answer is 160 millisecond sequence. The inclusion of that does facilitate learning. And that establishes another line of this triangle, i.e. replay does support and facilitate non-local learning. Okay, great. So the last question is replay actually prioritized so that it's going to most likely to replay the most policy informative behavior? Again, this can be translated in the analysis, i.e. do I replay more to the yellow path or to the green path, depending as a function of the product of lead and gain, right? And again, just to remind you, the lead is manipulated by the starting probability of the arm, and the gain is manipulated by the reward probability of each outcome. And then at each of trial, we can separate the high priority with the low priority. And when we do that and collapsing all trials, we can detect a difference in the replay that is stronger in high priority paths versus low priority paths. And so And we can see that again peaked at 160 milliseconds, while this 30 millisecond thing, which is dotted line here indicated by this blue line here, is not significant at all. So this establishes the last line of this triangle, i.e. replay is indeed prioritized. It prioritized so that it first replay the most policy informative experience. All right, with that, I want to conclude. So we find non-local learning is prioritized to support model-based behavior, and we find reverse non-local replay at 160 millisecond lag help model-based learning. And it's both functional and physiologically different compared to this Shepwave repo faster 30 millisecond replay. And this 160 millisecond replay is prioritized for the most policy informative experience. And with that, thank you, and thank you all my collaborators, Kim, Nathaniel, Marciano, and Ray, and thank you.
Hello everyone, I'm Xuebing Li. I'm from Institute of Psychology in China. First, I would like to thank my teacher, Professor Luo, invited me have this presentation here, and thank Professor Qin organized this symposium. Today, I will introduce a new type of working memory training devised by our team that aims to improve anxiety. We're all familiar with working memory training. It's a very popular training based on the computer. This working memory is implicit in many advanced cognitive functions. Working memory training can cause potential file transfer effects, such as increased reading comprehension, mathematics reading, fluid intelligence. Besides this cognition transfer, I am more concerned about could working memory training transfer to emotional regulation, and what might cause such transfer effect. There are two hypotheses underlying this relationship between the emotional regulation and working memory. There are essential overlapping brain regions have been reported involved in emotional regulation and working memory, such as dorsolateral prefrontal cortex and the parietal cortex. And also, working memory and emotional regulation have similar cognitive processing, such as updating, inhibition, shifting, and other processes has been talked by Badley and Grass a lot. So based on this hypothesis, there is already evidence to show that linear working memory training can improve emotional regulation when participants are watching negative materials. Even it can improve overall emotional states such as anger, depression. But from working memory to emotional regulation is a very far transfer. Fast transfer effects are thought to be small effects at best. So if you want to get more emotional benefits, you had better put some emotional components into working memory training. That has been proposed by some researchers. Swedger and his colleagues show that working memory involving negative faces and words as such as updating stimuli, rather than training that use some shapes, numbers, can improve the effective controllability of participants, which will augment the transfer effects of working memory training. So based on these results, we can see the benefits of two types of working memory training may point to different forms of emotional control or emotional regulation. Here is the model of emotional regulation proposed by Akin, Booth, and Grass. They divided the emotional regulation into two different types. model-based regulation, which is also called as explicit regulation, and model-free regulation, which is called as implicit regulation. Model-based emotional regulation uses explicit strategies such as reprisal. And as we have mentioned, many studies have proved the reprisal and working memory share the similar brain network, such as frontal parietal control network. So according to this, we hypothesize that the general working memory training may be more likely to cause the transfer of explicit emotional regulation improvements. And another type of emotional regulation is model-free. It's a type of implicit emotional control depend more on automatically conflict monitoring. What is automatic conflict monitoring? Actually, this function is usually detected by emotional strobe task. So when you're thinking about the emotional strobe task, you will see the participant need to finish this job. They will inhibit the distractor from emotional stimuli. So if we put some negative emotional stimuli into the general working memory training, we will expect that the participant will inhibit this emotional distractor. And after a long training, the participants' negative inhibition control ability will be improved. So this is our hypothesis. To do so, the main subject of this study was investigate the transfer effects of working memory training and emotional working memory training on explicit emotional regulation and implicit emotional regulation in anxious individuals. We also think the anxious individuals can take advantage of such training because many studies has reported they have impairments in both emotional regulation and working memory functions. Here is the way we choose the participants and training the participants. All those participants were screened from almost 1,000 university students in Beijing with trait anxiety scores. The cutoff score is 50. Finally, a total of 120 participants were initially recruited and randomly divided into 3 groups: working memory training group, emotional working memory training group, and waiting list control group. After some reason, they are dropping out, and finally 98 completed the whole experimental procedure. This figure showed the task we used to train our subjects. We designed a new type of dual dimension unback training application on smartphone. The working memory training task required participants to remember the color and the position of this colored block, and adjust the color and the position of the current block was the same as the or different from the ends forward character. Yeah, is the general and back task. In this emotional working memory task, you can see all the colored blocks have been embedded with negative faces. But the same rules and operations has been required for the participants, which means you don't need to remember the faces. You just need to remember the color and the position in this training, in this emotional working memory training. Here is the task we used in pre-test and post-test evaluations. Yes, there are many evaluations we have done in these studies. And after 21 days APP training, you can see significant cognitive and emotional benefits in both training groups. Both training groups showed increased working memory capacity. And you also can see the anxiety and the stress score of both training groups decreased significantly. This figure showed the positive refocusing scores for emotion regulation increased in both training group. But all these effects you can see in control group. We used this task to test explicit emotional regulation. It's very common task to evaluate the implicit emotional regulation. You can use the reprisal strategy, or you can distract your attention to background of the picture, and you will see this picture passively. In this task, we choose the LPP as the indicator, which is the very classic ERP component reflecting the neural response of emotional regulation. We can see the changes better in this histogram. LPP amplitude for appraisal at the post-test was significantly lower than at the pre-test for working memory group and emotional working memory group, but this was not observed in control group. So this result suggests that the both working memory group improved the emotional implicit emotional regulation ability. And we use the facial strip test to test the implicit emotional regulation ability. In this task, you just ignore the facial expression and judge the color of this face. And we choose the P3 as the indicator, which is represented attention allocation resources. Higher amplitude of P3 means you need to put more attention resource in the task. And the lower P3 represent the better inhibition control ability. So you can see only individual in the emotional working memory task group showed a significant reduce in these components of ERP. These results suggest that the only emotional working memory training can improve the implicit emotional regulation. I'm sorry for the noise. Also, we can see many correlations between behavioral and neural indicators, such as LPP reduction for reprisal can predict the increased positive refocusing for emotional regulation in both groups, in both training groups. But only in the emotional working memory training group, you can see the P3 reduction can predict the reduced negative bias scores of participants. Finally, we can make a very short conclusion. Our hypothesis has been proved. Working memory training can improve the explicit emotional regulation ability of anxious individuals. While emotional working memory training adding emotional distraction can further improve the implicit emotion negative control. So this new type working memory training can be a better choice to use in clinical application. Okay, that's my presentation. In the end, I will acknowledge some of my students, Han Zhongyi, De Likhe, Liu Yunqian, and Wan Yu. And thank you for all your attention. Thank you.
Hi, I'm Haiyan Wu, a psychologist and a neuroscientist trying to understand how our brain works in social context. Today, I'm going to share our recent work about how brain signal when people are perceiving other people can predict social conformity in face perception and following decisions. I will first introduce people have a very well-developed social brain that can adapt to social influence in daily life. And then we asked whether there's a negative bias in this conformity. As a new social neuroscientist, we asked EEG indicators of social conformity in face perception and test how individual variations predict social influence impression changes and the following decisions. Face perception, as critical aspect of social perception, the ability to recognize faces and the emotions and the social cues in faces as a central ability of social cognition. Our impression to others is flexible and changeable, which would be differed between people and affected by others' evaluations. So let me give you an example. Here is my photo. Just imagine you see my photo like this, and you may quickly have a first impression about me. So here is my face, and here is another, for example, the object. So what are the neural correlation of face perception when others look at it? From Cognition and Johnson's work, brain activation for faces appears to be more focal and more face selective from the childhood to adolescence. Evidence have shown that FFA as the most face selective area is here. Then imagine that if I post my photo to the social media, and you might get like judgments from others. For example, we might get 19 likes in Facebook. Probably we may get like some likes from in Twitter. And then those kind of evaluation or judgments from even from strangers might affect our ratings to the face. So with the face attractive task, we found that people change their ratings along with the peer pressure. Rosen and Rosman proposed the inferential hypothesis that people usually attribute larger importance to negative information than positive information. Therefore, it's reasonable to speculate that negative evaluation from others are more prominent or salient, which can lead to behavioral adjustment toward a negative direction. But is that true? Actually, there's some but not consistent evidence. For example, from questionnaire evidence that with this face task, they do find that people adjust more for when the group rating is more negative than when the group rating is more positive. The same phenomena has also been observed observed in Nook and Zakit study with even like food task that people would like to change their preference to the food to be like more negative ratings if the peers ratings is lower. EEGs, for example, 170 component has been utilized to investigate the time course of emotional phases processing. The frontal M400 has also been interpreted as an indicator of conflict processing, which can predict the rating changes in conformity tasks. However, in our current work, we are interested in how the variation in EEG state during the independent phase perception task can predict impression change and following decisions. And this work is led by my PhD student, Haoming Zhang. To investigate this question, we select two phase datasets from the pre-experiment rating task and got two sets with different rating ranges. In the phase set 1, we include two levels of trustworthy phases, low versus high. Half of them are males. And for phase set 2, we select 40 middle level trustworthy phases. So basically, this is a 4-step study. The first step is the initial phase judgment task with high variance. And the step 2 is the low variance stimulus with social inference. The step 3 is independent phase rating like judgment task with the same phase set 2. And then we have the step 4, the trust game with the phase data set 2. So here is the first initial phase judgment task with this the phase set 1. We print these phases and asked the participants to make binary choices like trust or untrust. And then we will go to the step 2. We manipulate 4 levels of trustworthy ratings from others. For example, for condition 0, it means that the red lines means that all the 3 judges this phase as untrustworthy. For condition 3, it means that all the 3 judges this phase as trustworthy. And the task looks like this. First, the participants see the photo and make binary judgment, and then the peers rating are shown. After this social inference task, participants made their judgment without peers ratings in the experiment 3 with the same phase set 2. And then we will go to step 4, is a trust game with the phase set 2 after the social inference. So participants make a trust game decisions with these virtual people from phase set 2. For example, we can imagine that after social inference, people are more likely to trust the phase with positive peer evaluations. So here we do get some results. First, we do find that people are more likely to change toward negative social inference. See condition 0, that all the 3 other people judge this phase as negative. So we can see that there's a significant behavior change. For the behavior change was defined by subtracting the average trust rate in experiment 2 from average trust in experiment 3 for each condition. For experiment 4, the behavior change or the trust rate is defined by subtracting the average trust in condition 0 and 1 from the average trust in condition 2 and 3. So we can see there's a very significant effect for condition 0. So, and now I will talk about the pipeline we used to analyze the EEG. It's like about how do we define the temporal EEG variability in experiment 1. For example, we use alpha band path filter and use time window to extract abundant temporal information. Within each time window, EEG variability is obtained by one subject, the average correlation. We have adopted this same measure to address the temporal variability of each subject participants and each electrode. In the end, for each subject and each electrode, we got a 1-dimension line. for EEG signal in experiment 1, we use all trials, like with both trust and untrust trials, to calculate the variability, because we do not find significant difference between trust and untrust phases in this experiment. And then for the social inference EEG in experiment 2, we calculate the EEG under different inference conditions and analyze this separately. So here is the temporal EEG variability average between all subjects. We chose 2 electrode and 2 frequency band as example. So the left side is calculated by the unfiltered EEG signal. The right side is actually calculated by the alpha band pass field EEG signal. So the 2 figures above are from AF4 electrode. The below are the P7 electrodes. The red line is calculated by all events in experiment 1. The green line is calculated by trust event. The blue line is calculated by untrust event. We use permutation cluster test to calculate the significance of the signal relevant to the baseline. The broken line means non-significant. The full line means significant. We can see the difference between the 3 lines are almost non-significant. So in our following analysis, we use all event variability to predict the behavior from experiment 2 to experiment 3. And also we use this variability to predict the behavior change in experiment 4. So as we can see, the right frontal alpha band can predict the behavior change from experiment 2 to experiment 3. For both negative social inference condition, we can see that if the others, like most of the others, judge this phase as negative, so the EEG signal or the alpha band of the right frontal lobe can negative predict this behavior or rating change from experiment 2 to experiment 3. And here are the animations, which shows that temporal change, that the prediction effect is stable from 60 milliseconds. Yes. Interesting, we have also observed a similar predicting effect for trust rate in experiment 4, but it's positively prediction effect. Okay. And also, we also found that frontal alpha band can predict the behavior change from experiment 2 to experiment 3. What does this mean? It means that this EEG signal is extracted from the segment, the epoches that when people see the peers ratings. So it means that all the frontal alpha band over the frontal electrodes can predict the behavior change from experiment 2 to experiment 3. It's kind of a serve of the EEG indicator of conflict in social that can predict social conformity. So from this animation, we can see this positive prediction effect from right alpha band to the trust rate of experiment 4. So here are some take-home messages. The first, that the negative social inference is more pronounced in face perception because we do found this effect. And also we found the right frontal alpha band and also the left occipital EEG in face perception can independently predict susceptibility to social influence in this face perception task. And also we found that individual EEG variations in the independent task can predict both the social influence impression changes and also the following decisions to certain phases. Okay, thanks for your attention and thanks to my PhD student Haoming for working on this project. Our lab is recruiting PhD students, research assistants, and postdocs. So feel free to contact me if you are interested in social neuroscience. You can also visit our lab to know more about our lab. Thank you.
Hi, I'm very happy to present at the World Congress of Psychophysiology. My name is Frederik Weber. Today, I'm going to present about Sleep Trip and other open source tools to analyze sleep data, polysomnography data, especially in clinical samples. I will use major depressive disorder patient data as an example. And I'm affiliated with the Donders Institute in Nijmegen in the Netherlands, together with Professor Martin Dresler, as well with the Netherlands Institute for Neuroscience in Amsterdam, also the Netherlands, together with Eus van Zomeren. I have nothing to disclose as a conflict of interest.
Obviously, in the recent years, sleep has beginning to get much more interest in brain research, and it's getting a larger and larger proportion of brain research, now reaching over 20% of brain research by the metric of just how many PubMed terms are associated with this. That means more and more data is created, and we can also see that from the accumulation of larger and larger national and international data sets that create a lot of sleep polysomnography or sleep EEG data. However, the problem is when we look at such sleep EEG data, we try to look at small things and not at the big picture. Here's a big picture as an example for what I'm going to talk about. So you can see lake brain, and if you want, we can measure some EEG from this lake brain and also infer some phenomena that we can commonly measure. For example, slow waves, spindles, or infraslow oscillations, or any other aspects of sleep phenomena that we can commonly see in polysomnography data, or also infer from the deeper inside the brain or the lake brain. However, if we look closely at every detail, we notice that we're actually not looking at a lake. We're looking at something false. It's something like a wall. And so far, recognizing that there is a wall, you need to look at a lot of different aspects of this big picture. So shall we tear it down? Is this possible? I suggest that's also possible with open software tools, and currently I'm developing one of them as an example that this is feasible. A commonly known tool for analyzing electrophysiological data is Field Trip, which has become in the last decade one of the most utilized tool to make sleep research pipelines or sleep analysis pipelines, as well as any electrophysiological analysis pipelines, because it's very flexible and accommodates to the common needs in this field. Recently, I've begun to make a branch of Field Trip that I call Sleep Trip, which is a full Sleep Trip version with added functionality, where I also integrated features of previous toolboxes to do some sleep analysis in a more pipelined way. And the purpose and goal is to have an analysis created quickly, to make this analysis replicatable and standardized, so it can be adapted by a large community of users, and it can be done in a familiar framework like Field Trip. Some features include from sleep scoring and evaluation, manually or automatically, the import, comparison, listing, or extraction of sleep cycles, cutting of that, conversion sorting, and so on. So dealing with sleep scoring, detecting specific events like slow waves, spindles, the power spectra and bands, co-occurrences, traveling waves, slow oscillation spindles or slow wave spindles, rapid eye movements and whole eye spectrograms, etc. Visualizing those in hypnograms, a data browser, topography, or other things that are commonly done in field trip. Also, this supports some pipelining tools, so processing and further processing, like integrating a pipeline, filter information in EEG, apply some grammar or montages to the data, aggregate results and export them so they can be dealt also in other software. Need to mention that this, since it's based on field trip, works on most common EEG and MEG standard formats that are used also in sleep research. If you want to take a look, here's my GitHub profile. You can just search for sleep trip and my name, and it should pop up where I have all those algorithms and the code available, as well as an example pipeline where you can see how that works. Then I applied this data. I applied this toolbox to a larger data set as a proof of concept that you can also deal with this in larger populations, in larger data sets, and also not just arbitrary data sets, but clinical data sets. It was a data set that was collected in Munich with the involvement of Axel Steiger, Marcel Zeising, Michael Zisch, and Martin Dresler, and which we analyzed together, Dr. Leonore Wovi and we, to include those features on a big picture level of what happens during non-REM sleep in major depressive disorder. Here you can see we had 3 different data sets, data set A, data set B, and data set C, where they all have varying degrees of depression. For example, here expressed in the Hamilton score from moderate to severe depression, and also in different states, from being in a medicated state or being in an unmedicated state in, for example, data set B, or having a medicated state with a follow-up for 7 and 28 days later. So this allowed us to check if there's minor differences in some of those sleep features between patients of severe to moderate depression under medicated or unmedicated state compared to age and gender matched controls. What we surprisingly found is that only in the medicated state we see major differences that are commonly referred to in the literature as disruptions in the sleep architecture for depressed patients. However, we, to make it short, we didn't observe any major differences except slightly increased wake after sleep onset time in unmedicated patients of moderate depression at a young age. So also we did not find that there's any alteration of the slow wave sleep, at least on a macroscopic level or the scoring level. We also did not see that there was a major disruption of REM sleep, in particular for the percentages, if it was not medicated in a specific way. Therefore, we looked further into non-REM features to check if there is some microarchitectural changes, specifically depending on the medication. Don't want to go too much into details. If you want to take a look, please go to the publication. It's on Biomarkers, easy to find, and was published just recently. So as you can see here is that the duration of the sleep cycles have increased, which was not surprising. So if the patients were medicated compared to the controlled, they had longer sleep cycles, which was mainly limited to the longer non-REM sleep cycle, which was observed in each of those data sets, but only under the medicated state was significantly altered. And this could be traced down to medication that was also known to be REM suppressing. So suppressing REM and prolonging non-REM. So that fits the general picture. So that highlights the how crucial it is to really check for controlling of medication. And I know that in a lot of samples and previous samples that have been reported, they have not, most of the subjects, even if they have been unmedicated for the studies, have taken medication at least a week or 2 weeks in advance and just abstained for that for some period, which might still have some long-lasting effects, as I might come to that later. Also, as a comparison, if we just look at the hypnograms here of all the younger subjects versus all the older subjects of the complete data set, one can immediately see that, for example, here marked in red, that the younger subjects in the first sleep cycle, they have more pronounced slow wave sleep. And in the older subject, that becomes more fragmented in general and is less present. However, this data is now sorted by the length of the first sleep cycle. One can see that the overall structure of how long a REM sleep cycle is not so much altered between young and old, where we know there's a lot of effects on sleep EEG and sleep architecture. However, as a comparison, if you look at the medicated patients versus healthy controls, just an overall look, it becomes immediately apparent that the structure in general has changed, as you can see here with the healthy controls having a rather normal sleep cycle length on average with not a large variability. However, with medicated patients, that the REM cycle, either due to early REM onset or to very prolonged or a variable first sleep cycle length, this shows that this might be very much disrupted. So just as a comparison, so those medicated effects might even, at least in sleep architecture, be more obvious than the age effects in this data set. Further looked also into just the spectrum of non-REM sleep and the power spectrum. So checking if there's some differences in a certain kind of oscillations. And there is on medication status a difference in a reduction in slow wave activity, which we could later also trace down to the slow wave amplitude in those data sets that have been long-term medicated, not just for 1 week, but typically for longer, where we see amplitude reduction. For spindle activity, there was also with long-term medication, which is also indicated in this data set, and for the specifically controlled medicated 28-day data set, that there is a reduction of spindle density overall. We also looked at slow wave spindle coupling. That means spindles that really are in the upstate of a slow oscillation. We didn't see any differences in count or the delay in which that happens with respect to the downstate. However, we see that there is a difference in how variable this delay from the spindles to the downstate is. So that means this delay dispersion is typically increased under medication or longer term medication, specifically the older the subject are, meaning if these subjects of the data set B, which are generally a little bit younger and have had less episodes of depression, we won't see this so frequently. There's this. So since in the light of the symposium, I also want to use the opportunity to talk a little bit about the aspects of how to do stimulation experiments and how to make this feasible. At my heart is that research should be available to a large audience of researchers, not just for institutes like we are in that are privileged and have typically high-end equipment, but it's also good to have like field work or allowing research somewhere else. And commonly there is a trend to having just some home equipment and being able to use inexpensive open source hardware to record sleep, and I've done that in the past as well. And you can even build an uncomplicated full PSG device, which I jokingly call somnophredics, which is rather inexpensive but works really well. And the data quality is also excellent from my view. You can do a full PSG setup. And most importantly, I also developed a software that works directly with this hardware to record full PSG data and the different montages. And furthermore, you can also do a stimulation experiment. For example, auditory closed loop stimulation during sleep, or target memory activation due to sounds. So for example, you can create with the software a closed loop or an open loop protocol for stimulation during sleep. For example, closed loop locked to the slow oscillation down state or to some part of that, and then targeting a specific interval after that if the signal falls in a specific range and so on. With this, you can reproduce very easily some protocols that have been published before, basically nearly all of them that are with auditory stimulation at least already, and with slow oscillation. So, and I'm currently also extending that. It's already publicly available. You can find it on GitHub. And for now, it supports OpenBCI as a software, and also will support some other software in the future. Here's a proof of concept. There's some pilot data from some time ago that shows that such evoked responses can be easily created with this software, that you have an auditory cue that comes after slow oscillation that also creates an auditory evoked potential like you would expect. And you can also see in other research that has been recent. So the concept works. And this was pilot data from a collaboration of the Beijing Normal University with the Donders Institute in the groups of Professor Xiaocheng Jing and Professor Martin Dresler and collaborator Xingyi Wang and the team there. So obviously, I have also some memory data. If you're interested in the questions, can go into further details about those data sets. But other than that, I want to acknowledge also Robert Ostenfeld and Jan Matthäus and the whole field trip team that supported Sleep Trip very well, and also people that already use Sleep Trip and the other toolboxes that I created. Thank you for your time. 谢谢. And that's it.
Thank you for inviting me to IOP 2021. My name is Wei Wu, and I'm currently a professor in the School of Automation Science and Engineering, South China University of Technology. It's my great pleasure to be able to have this opportunity to share our recent progress on EEG signal processing and biomarker discovery in an effort to achieve precision mental health. I have been working with EEG for many years, and I'm still fascinated by how much information we can get out of EEG and how much we can push the limit of this classic brain recording technology in translational research. So here is an outline of my today's talk. I'll begin with the challenge in precision mental health. Then I'm going to walk you through our recent EEG biomarker research conducted to address these challenges. Finally, I'll briefly introduce our ongoing study aiming to optimize brain stimulation therapies via EEG biomarkers. Psychiatric disorders are leading contributors to the global disease burden. In particular, depression incurs $100 billion per year in healthcare costs, and PTSD accounts for 60% of VA's health spend. Despite such an enormous spend on mental health, there is actually very little progress that has been made in treating psychiatric disorders. The fundamental issue is that psychiatric disorders are diagnosed based on combinations of symptoms, but patients with the same symptoms may actually fall into biologically heterogeneous subtypes, which may require different treatments. However, to date, there is no validated biomarker that can reliably dissect such biological heterogeneity and allow us to objectively diagnose, track, and treat psychiatric disorders. The entire process is fraught with subjectivity and trial and error. There are two predominant approaches for using neuroimaging data to parse the heterogeneity of psychiatric disorders. The first approach attempts to derive biological subtypes using unsupervised machine learning based on neuroimaging data alone without referencing to clinical information. The derived subtypes are therefore more likely independent of treatments. By contrast, the second approach maps the pre-treatment neuroimaging data to the symptom change as a result of a particular treatment using supervised learning. This approach therefore yields treatment-specific biomarkers and is able to predict whether a patient responds or remits to the treatment. So next, I'm going to tell you two representative biomarkers we recently discovered using these two approaches, and each discovery entails methodological innovations in EEG signal processing and machine learning. In the first biomarker study, we were able to discover transdiagnostic subtypes based on EEG alone. The EEG feature that's employed to derive the subtypes is the power envelope connectivity between different brain areas, which captures the correlation between slow power fluctuations of the EEG oscillations. In an earlier study, we have shown that EEG power envelope connectivity is able to yield brain networks similar to fMRI connectivity, demonstrating it is a neurobiologically meaningful EEG connectivity metric. Based on the power envelope connectivity, we developed an EEG-based subtyping framework for psychiatric disorders. Specifically, the resting EEG data is first filtered into different canonical frequency bands. The power envelope connectivity is then calculated between different vertices in the source space. The vertex level connectivity is then averaged within each pair of brain regions to yield the RI level connectivity. Considering the high dimensionality of the connectivity features across frequency bands and resting eye conditions, we employed a sparse clustering algorithm for simultaneous feature selection and clustering, as opposed to conventional clustering algorithms, which typically put all the features on an equal footing, and therefore these conventional algorithms are not able to determine the clusters correctly when some features are redundant or noisy. But in this case, the sparse clustering algorithm is able to automatically zero out redundant features and select informative features as the dimension to perform clustering. This subtyping framework allows us to derive subtypes in a fully data-driven manner. Applying the proposed subtyping framework to the resting EEG data of a PTSD study revealed two subtypes with distinct connectivity patterns. What I've shown here are the connectivity matrices for healthy controls and the two PTSD subtypes. Between the two subtypes, the connectivity patterns are highly similar between subtype 1 and healthy controls, but are very different between subtype 2 and healthy controls. This observation is confirmed by calculating the correlation coefficients between groups. The sparse clustering algorithm was also able to select features predominantly from the eyes open condition and the beta band as the main driver for the two subtypes. Performing t-tests between the connectivity patterns of the two subtypes suggests that subtype 1 has significantly stronger connectivity within frontal regions, but weaker connectivity within parietal and occipital regions. The interhemispheric connectivity between homologous regions is also notable. To test whether our discovered subtypes could be replicated, we applied our subtyping framework to resting EEG dataset of a second PTSD study. Unlike the first dataset, which was collected in a cross-sectional study where there's no treatment involved, patients in the second dataset received the psychotherapy treatment after their resting state EEG data were collected. And it's worth noting psychotherapy is considered as the gold standard treatment for PTSD. We separate this dataset into two cohorts. The first cohort, containing 72 patients, was used during the initial submission of our paper. And the second cohort contains 63 patients who completed the study after our initial submission and was used for further independent replication of our study. In the first cohort, our subtyping framework again yields two subtypes with differential connectivity, which is consistent with the results from the Discovery PTSD dataset. Since patients received the treatment in this dataset, we are able to assess the clinical significance of the subtypes by examining treatment outcome associated with each subtype. Using linear mixed models, we found that subtype 1 is more responsive to psychotherapy in terms of both CAPS-4 and CAPS-5, which are two versions of clinician-administered PTSD scales. Similarly, in the second cohort of the second dataset, our subtyping framework yields subtypes with patterns consistent with those in the previous data. The subtype status again predicts responsiveness to the psychotherapy treatment, with subtype 1 responding significantly better than subtype 2. We next investigated whether the discovered subtypes could serve as a potential transdiagnostic biomarker. To do so, we applied the subtyping approach to an independent depression dataset collected in a placebo-controlled randomized clinical trial where patients received either antidepressant or placebo. Quite strikingly, once again, two subtypes were observed with distinct connectivity patterns consistent with those discovered in the two PTSD datasets. Moreover, linear mixed model revealed that the two subtypes differed in the response to antidepressant versus placebo. Specifically, while antidepressant was significantly better than placebo in subtype 1, the two arms failed to separate for subtype 2. The treatment response analysis also revealed that subtype 1 has significantly more responders than subtype 2 for antidepressant. However, for placebo, there is no significant difference in the number of responders between the two subtypes. Now, the first biomarker was derived cross-sectionally without referencing to any particular treatment. In the second biomarker study, we discovered a treatment predictive EEG biomarker for depression. Conventionally, EEG machine learning methods use a divide and conquer type of a strategy to do treatment prediction. Specifically, to mitigate volume conduction, they first optimize a set of spatial filters, essentially by providing weightings on different EEG channels and combining them to yield the latent signals. And then the band power feature of the latent signals is calculated, followed by linear regression that relates the band power features to the treatment outcome. The spatial filters can be optimized using unsupervised approaches such as PCA, ICA, or even source localization. The linear regression model can be optimized with ordinary least squares or sparse approaches such as lasso. However, the major limitation of these approaches is that the spatial filters are optimized with cost functions unrelated to treatment prediction, and therefore these algorithms are not optimal with respect to treatment prediction. To address this limitation, we developed a new machine learning approach called SELSER for EEG-based treatment prediction. Unlike conventional machine learning approaches, in SELSER, the spatial filters and linear regression model are no longer optimized separately with different criteria. Rather, they are now optimized in conjunction under a unified framework, which is minimizing the prediction error. Importantly, this approach is able to handle several important challenges. The spatial filters are optimized in a supervised manner to ensure optimality with respect to treatment prediction. The number of latent signals are minimized via low-rank regularization to avoid overfitting. The optimization problem is formulated as a convex optimization problem such that the global minimum solution is always guaranteed. We apply Seltzer to the pre-treatment resting EEG data from the Embark study, which is so far the largest neuroimaging coupled clinical trial for depression. We found an EEG signature that's predictive of the treatment outcome for sertraline, which is a widely used antidepressant, with a correlation coefficient of 0.6 between the true and predictive symptom change. Applying this signature to the placebo arm doesn't predict the symptom change, suggesting that this EEG signature is specific to sertraline. The spatial patterns of the latent signals are predominantly in the parietal occipital regions and the frontal regions. Now, Embark is a multi-site study with different EEG amplifiers across 4 study sites. This allows us to assess the robustness of the algorithm to different EEG systems by performing the leaf side out prediction. To do so, we trained the model on the EEG data from 3 study sites and then tested it on the EEG data of the remaining site. In this arguably worst case scenario with different EEG systems between the training and test sets, Seltzer is still able to find a sertraline signature predictive of the treatment outcome. We also validated the sertraline signature on independent depression datasets. The second dataset is from a naturalistic study where historical information regarding treatment resistance to antidepressants is available. By applying the EEG signature to this dataset, we found that partial responders have significantly greater predictive symptom improvement than the treatment-resistant group. This finding therefore demonstrates the generalizability of the EEG signature to the broader construct of treatment resistance to antidepressants. In our earlier study, we also established a task fMRI predictive signature for sertraline in Embark. To test the convergent validity of the EEG and fMRI signature, we applied them to another depression dataset where patients were assessed in a cross-sectional manner using both resting state EEG and task fMRI. On this independent dataset, we found the resting state EEG and task fMRI predictions were significantly correlated with each other. This finding provides convergent support for the existence of a treatment-responsive neurobiological phenotype in depression across neuroimaging modalities. Finally, I'd like to use this one slide to talk a little bit about potential future work. The discovered biomarkers alone do not directly inform therapeutic development. To drive therapeutic development, it'll be helpful to develop methods to identify causal circuits associated with the discovered biomarkers, which may provide new targets for brain stimulation therapies. Specifically, we may use concurrent TMS EEG to stimulate multiple nodes in the brain and then relate EEG biomarkers to the concurrent TMS EEG maps to be able to identify the causal circuit abnormalities. The identified causal circuit can serve as the target for repetitive TMS to remediate the circuit abnormalities. Studies like this may yield a principled approach for us to develop brain stimulation therapies based on the EEG biomarkers. I'd like to thank my longtime collaborators and co-authors for the tremendous help on my research and various funding agencies for supporting my work. With that, I'll stop, and I'm happy to address any questions towards the end of the session. Thank you.